Okay, so I'd like to welcome everyone to our last mini seminar and working meeting in January of the IMAG MSM Modeling and Pandemic Working Group. We have a very exciting speaker lined up, Niarakis from Paris. As usual, I need to remind you that this meeting is currently being streamed, it's being recorded, and the recordings will be slightly edited and made publicly available. So please don't disclose proprietary information or other things that you don't want to be shared. I remind you that I'm a co-lead, James Glazier, along with Reinhard Labenbacher, who will be joining us in a few minutes. We have support from Jim Sluka, who has been doing all the back office work, and a lot of you have talked to. And you've certainly heard from Bruce Shapiro on scheduling, emails, and things like that. And we appreciate their help, and you can contact us about any issues related to the working group. As always, I remind you we have a Slack channel. It only works if you use it. Please feel free to use it. We also remind you that you're welcome to post material on the IMAG MSM wiki page. We'd certainly encourage the working group subgroups to do that. And please feel free to correct things that are incorrect and suggest additional content. For upcoming meetings and mini seminars, we are lucky to have Morgan Craig and Greg Forrest talking next week. Morgan will be talking on in silico trials, and Greg will be talking about viral entry and transport. The following week, we have 2 talks lined up, and there are intermittently scheduled talks after that. As usual, we would love to have your suggestions for future speakers. Don't be shy about suggesting yourself as a speaker. We are always open to suggestions, and people certainly don't have to be working group members to speak. And we're probably a little early to have people speaking a second time, but those of you who've spoken once and didn't get through the material you wanted to present, have new material or other topics, are welcome to present again. I always try to keep this preambles as short as possible. So I'm going to turn it over in just a second. We're very lucky to have Anna speaking today on COVID disease map, computational knowledge repository. And with that, I will turn it over to her.
Okay, so thank you very much for the invitation. I will start sharing my screen. I hope that you see my screen, and if everything is okay, then I will start the presentation. So today, the goal of the presentation is to give you an overview of the COVID-19 disease map project that is a large-scale community effort in order to build a computational repository regarding virus-host interaction mechanisms. So the goal of this consortium, of this group, is really to apply systems biology approaches in order to achieve a full integration. So the key goal of the disease map project is to construct formalized and standardized mechanistic networks at different levels of granularity in order to investigate cellular machinery and host pathogen interactions. So we would like to use this repository as a template, as a help, as a support to interpret large-scale data sets, extract true information, and understand how the infection takes place, propagates, and escalates. We would also like to develop, and we do so, we develop computational techniques for integration of large and heterogeneous data sets and combine them with prior knowledge. And last but not least, we build computational models that can provide insights into mechanisms of interest. So what do we offer as a community? We offer network encoded disease-specific knowledge. We offer templates for omics data analysis depending on the data sets that one might have. and provide a means to derive molecular, cellular, or patient profiles. So these type of mechanisms of disease representations can be used as templates for drug targets and for drug repurposing analysis. And also we offer methods in order to derive executable models. We use literature and information mining, biocuration, human-based biocuration, but also we integrate assisted biocuration from artificial intelligence solutions. And we are advocates of systems biology standards in order to facilitate interoperability and be in line with other community efforts. And as you will see, our community has a broad range of expertise. So we are also relying on experts' feedback in different aspects of the project. So these are numbers of last month. I don't know if we have grown bigger since. So we have around 267 members from 128 institutions and 31 countries. These are information. This is information derived from our Ferdom Hub page. The expertise is really broader. So we are a community of clinical researchers, life scientists, computational biologists, bioinformaticians, and most of us come from academia, but we also have collaborators from pharma companies and from small to medium bioinformatics enterprises. So for the communication between us, we use a web page, the Ferdom Hub page, Google platform, and we also host weekly theses. Every Friday, we host two theses, one in the morning Paris time and one in the afternoon to accommodate people in different time zones. We alternate every Friday between curation theses and data analysis and computational modeling. The biocuration teleconferences are hosted by Marek Ostaszewski from the University of Luxembourg, and data analysis and computational modeling are hosted by me. So this is our page in the Ferdom Hub. There you can see that there is a certain transparency about who contributes, who is doing what. Data files, models, publications, and documents that we use that can be accessible by everyone who participates in the project. So regarding curation, we share interesting COVID-19 articles via Zotero group. This group is public by invitation. So if you would like to be part of it, just contact us. We have curation guidelines because, as you will see, many different groups around the world actually came together to curate and build mechanistic diagrams in a standardized form. So we had to give some curation guidelines and some criteria in order to have harmonized graphs that respect systems biology, graphical notation, and modeling rules in general. Regarding the content repositories, all the curated diagrams and all the code and scripts that we are using are placed in a public GitLab repository. Access, you can have access with your GitHub login, and if you want developer privileges, you just contact me or Marek. We also collaborate with Wikipathway and Reactom. Wikipathways can be shared using the dedicated collection of the platform, and Reactom has released two SARS-CoV-2 infection diagrams for SARS-CoV-1, and SARS-CoV-2. And there is also a weekly builder of the platform-independent diagrams that the community is building. So to give some more insights, we are mostly using process description and activity flow diagrams. So all the content that is curated and translated into mechanisms respect systems biology notation schemes. So in the community, the from scratch mechanisms are built using process description, but in platforms where we collaborate, like the Wikipathway collection, activity flow is privileged. Regarding Reactom uses also a process description notation scheme for the diagrams. So this is just to show you that we call it the COVID-19 disease map, but it's really a collection. So there are 21 diagrams that we are hosting in the Minerva platform, as I will show you in a short while. We also have Wiki Pathways, 19 diagrams in the Wiki Pathways collection, and also 2 large diagrams in Reactom. So instead of building one large map that would connect signaling, gene regulation, and metabolism that would actually be chaotic in terms of scale and also collaborative efforts. All these groups focus on mechanisms of interest where they have a certain expertise, and they use this expertise in order to translate their knowledge into SBGN diagrams. So this is a top-level view of the project. We have a human host cell, and then we have the respiratory tract, the systemic circulation, and tissue organs and system. And we have different mechanisms that can be put in different parts in this top-level schema. So most of our mechanisms are here in the host cell, type 2 pneumocyte, so, and also in the adaptive and innate immune response. We host the platform independent, let's say, besides Wiki Pathways and Reactom, we host all the new mechanisms in Minerva. That is a platform developed by the University of, by the Center of Systems Biomedicine in the University of Luxembourg. And Minerva is a platform for interactive visualization of the content. So it looks like, if you zoom in, it looks like a Google map. It uses this type of technology. There is a search box where you can search interactions or molecules, for example, interleukin-6. And Minerva will tell you in which of these subgraphs this molecule exists. You can zoom in, for example, like this. You can see that the molecule that we were searching in this example, it would be the ORF7 alpha. It is in the diagram of virus replication cycle. You can continue to zoom in, and then you actually have access to the SBGN diagram. You also have access to all annotations that the curators have included in the diagrams. And you also have, for example, PubMed IDs, taxonomy, and everything that was provided by the curator. But there is also an option to have automated annotation from databases. So the areas of focus are curation and content sharing, integration of different types of information and graphs, and also formats in order to foster interoperability and exchanges between the community. And last but not least, bioinformatics analysis pipelines and computational and mathematical modeling in order to derive more dynamic and more mechanistic insights. So this is the ecosystem of the COVID-19 disease map, which has not been updated. We have 4 more tools that are now interoperable and part of the ecosystem. So as you can see, we have curators and the platforms that they are using. We have domain experts that give their feedback regularly in order to validate the mechanisms. And then we have analysts and modelers that are a little bit, let's say, more downstream of the first content. So there is a whole ecosystem of formats, interoperability, going back and forth, and things that facilitate, in some cases, text mining that goes back and forth with these platforms, but also analysis, simple analysis like topology that you can do with Cytoscape, or interaction databases like Senior that can be used in order to enrich the content. And then using different tools, you can obtain executable Boolean networks in SBML qual that can be used downstream to, let's say, as input to feed different types of analysis, as we will see in a minute. So there is really a lot of effort in the last year of many people who are working in the spare time, and they are volunteering for this project because there is no funding. So all these people have been working diligently for almost a year now to develop interoperable platforms and pipelines in order to contribute to the pandemics. So the first paper of this initiative was a short communication where last May we published it as an open invitation to the community, and the response was actually overwhelming. So from May that we had an open invitation, we published, we wrote the first manuscript that it's in bioRxiv and is going to be submitted for peer review on Monday in 6 months. And as you can see, we started with a handful of people, and here we have more than 200 co-authors. So we're building diagrams using Cell Designer, that is a popular diagram editor, and we make proper care. We make a lot of effort, and we take proper care in order to annotate properly and meticulously all nodes and interactions in the graphs using Miriam identifiers and proper qualifiers for every information that we stock in this type of graphs. So the human curation is high quality, but it's time consuming and laborious. So for all of these diagrams that I showed you and all these diagrams that you can see here, we have an average of 2 to 3 people working per diagram. So for 21 diagrams from scratch in Minerva platform, we used 624 unique scientific articles, and it took almost 6 months. So at first, it was hard to find literature for SARS-CoV-2, and most of the graphs were based on SARS-CoV-1 and other coronaviruses literature. Today, there is an estimation of 10,000 new COVID-19 articles per month. So you can understand that this is an information overload, hard to remain up to date, and human brain cannot really process all this information. So we need to help curators find the information faster, give some confidence scores, filter, help identify relevant content, and build efficient human-machine collaboration. So in the project, we collaborate with different groups and also small enterprises like Biomax Informatics that they have developed Ailani, a scientific assistant for COVID-19 research, where all the members of the community has free access, and it's really helpful in terms of facilitating curation efforts. We also collaborate with Peter Sorger Lab at Harvard Medical School, John Bachman and Benjamin Gyori, and we are using the intrasystem integrated network and dynamical reasoning assembler into the project. Intra is an environment where it can use information from various knowledge sources, pass this information into statements, into causal statements, and then extract this information, assembling them in different types of formats that can be used for different purposes. We also have, for example, just to give you an idea, here we have a graph of the COVID-19 disease map, and we have 251 evidence found for one particular reaction by intra. So there is a full alignment between curation, adding confidence scores, but also helping intra statements by filtering things that are irrelevant from the algorithm. We also have a bot. We have a Slack channel, and the intra people have developed a bot, the Claire bot, that uses dialogue, human-machine dialogue, in order to learn. So we are teaching the bot, and on the same time, the bot gives us back information. And this is a human-machine dialogue that can facilitate hypothesizing and discovery using background knowledge. So all these things are really nice, but up to here, we are, let's say, in a more static phase. All these graphs are very helpful as knowledge repositories and very rich in information and also in mechanistic information, but still they remain static. So we can perform topological analysis and highlight some key nodes as hubs, as interconnected, find some patterns maybe, but as you have seen, the COVID-19 disease map is not a global map. So topological analysis makes sense only in a, let's say, from a local point of view, per graph and not per mechanisms. So models are used to represent actual quantitative or qualitative relations between the molecules in a system by definition, and this we already have. They are also abstract representations of biological processes, and this is what we have also. But up to now, there is not an inherent execution scheme to make these models executable. So I have paraphrased a little bit Palson and the central dogma of biology. And in this approach, we have this dogma of computational systems biology that says that, I'm sorry, I don't know if someone would like to have a question. I see something. No, it's okay. Okay.
We usually hold questions till the end, but if somebody
Okay.
has an urgent question, that's fine.
Okay, I think I can go on, and we can keep questions for the end. So for us, identifying components is really important, but also it's very important to identify interactions, because this helps us build networks. And then we really need an execution scheme, a mathematical way to describe this type of interaction if we would like to have computational models that can help us perform in silico perturbations, in silico experiments, and study emerging behaviors and phenotypes, and then confront them back to real data, experimental data, and see what comes out. So prerequisites for a high quality molecular interaction map is that it needs to be accurate. So it needs to correctly represent empirical knowledge. And this is why we are using standards in order to avoid ambiguous ways of representing information. They also need to be reusable, because things that are not well annotated and well referenced, they cannot really be used outside of the community. When you find something that looks interesting for you, if you cannot find the source or justify what you see, you are going to redo the work. So this is why we are taking proper care for annotating properly all the material, all the content that we have in the repository. They also need to be comprehensive, that people understand what we would like to say, and machine readable, because after a certain scale, they cannot be processed visually, manually, or by the human brain. And last but not least, it's nice to have things that are executable, that we can test different scenarios, multiple perturbations and signaling cascades, and see what comes out at the end. And functional, because executable says nothing if the emerging behavior does not match the expected biological behavior for certain scenarios. So for this part, we have been using a tool that we have developed in collaboration with INRIA, that is the National Institute here in France for Informatics and Automation. CASC is a tool that uses cell designer graphs and converts them to executable Boolean networks. So what the tool actually does is that it takes process description diagrams and converts them to activity flows, and we also get a bonus of a preliminary Boolean rules, a mathematical description of the regulation based on topology, but also on semantics already encoded in the map. Semantics like activation or inhibition. So very briefly, the tool takes XML files as input and gives as output a valid SBML qual file that can be used with modeling software. And in terms of topology, in terms of structure, you see that from a process description that we have in the molecular maps, we will end up having activity flows through a series of steps and rules that the algorithm uses in order to infer structure plus the rule. So here is a simple example. This is the apoptosis module in the COVID-19 repository. It is an XML file. After using CASC, you obtain an activity flow. So you have a network compression, a regulatory graph, plus preliminary logical rules, Boolean, that describe the regulation. These files are completely executable, are fully executable SBML qual files that can be imported in simulation platforms like the Cell Collective developed by Thomas Helikar in the University of Nebraska-Lincoln. And as you can see, the rules are translated into conditions and you can play with them. Here, it's a very simple example. You set to 100% activity that corresponds to a Boolean one fast and fast ligand, that is a death signal. And then you ask to monitor what is happening to the apoptosis phenotype. So you can use this type of real-time in silico simulations to understand input-output relationships, and if your model actually is consistent with the biological behavior that you would expect. This type of models also inherit the layout of the original graph, which is very important when you have large models. Here is a toy model, but imagine that if you have 200, 400 nodes, knowing where your receptors and ligands are and where your phenotypes are also facilitates simulations. And lastly, CASC makes the translation in such a way that all the references stored in Miriam section of the graphs using the proper qualifiers can be retrieved in the SBML qual file and can also be retrieved in the modeling platform of Cell Collective. So what you end up here is having is models that are actually annotated both for nodes and interactions. So there are 21 SBML qual files in the GitLab repository and also 21 C files that describe the topology. SBML qual files are used for the executable dynamic modeling, but C files are also used for mechanistic modeling. We have two different types. So the first type is used by Hypatia, a model developed by Joaquín Dopazo at Seville that uses topology, activity flow diagrams, plus omic data, transcriptomic data, and produces mechanistic models for activated and downregulated pathways. The second C file is used by the group of Julio Saez Rodriguez that uses Dorothea and the pipelines developed by the group to assess transcription factor activity and then map this transcription factor activities, these profiles back to the graphs. At this point, we are focusing on the dynamical modeling of type 1 interferon. So this is a modular level modeling effort. And we have a model that describes type 1 interferon after SARS-CoV-2 infection. This is an SBML qual file with preliminary rules that now is being analyzed downstream with a set of tools that are compatible with SBML qual. We are collaborating with the Colomoto Consortium, the consortium for the development of logical models and tools. And Aurélien Naldi is making efforts in creating reproducible pipelines using Jupyter notebooks so that every simulation and analysis of this type of files is accessible, transparent, and reproducible by everyone. The group of Martin Kuiper in Norway has been developing a COVID-19 macrophage model. So what they have done is that they have taken 3 different graphs from the repository. They have used CASC to translate them into Boolean models, and then they have integrated these graphs, these Boolean networks, with a macrophage polarization model that they had been building in their lab. And last but not least, a very exciting, let's say, possibility is to link all these pipelines and all this context downstream with PhysiCell and PhysiBoss in order to perform multiscale simulations for the effects of SARS-CoV-2 on alveolar human cells. So how do we go there? We use PhysiBoss, and as you can see here in the diagram, we take the XML files from Cell Designer, we translate them to SBML qual files with CASC, and then these SBML files can be used to feed the Colomoto pipelines, for example, GeneSim. They can be imported into GeneSim, and from GeneSim, they can be exported as MABOS compatible files, and from MABOS, we can actually have compatible files for PhysiBoss, and this can be integrated with PhysiCell. So there is a whole cascade of tools that are suddenly accessible due to the use of standards and to the interoperability assured by the community. So to wrap up a little bit, the idea here is that we have Boolean models that describe the behavior of the cell, and they are plugged into the multiscale modeling of the virus. And here is the first attempt from the Barcelona Supercomputing Center and from Institute Curie. The first thing that we did was to build a phenomenological model between the virus, the replication, the apoptosis, the immune signaling, and then the crosstalk with T cells and macrophages. So we used this graph, the apoptosis module, CASC, in order to produce a Boolean network, and then we plugged it into PhysiBoss and start simulating different scenarios. For example, a knockout of FAD that is very important in the apoptosis cascade, and also played around with proportion of cells and what is happening if we inhibit the M protein of the virus. We are also trying to encourage people develop kinetic models. So we had one kinetic model, a parallel effort from the group of Andreas Drager at the University of Tübingen. So Andreas also has been developing tools like the SBML squeezer that can be that can facilitate the reaction kinetics, but these graphs are SBML in their essence, so they can be imported in Copasi, for example. But still, there is this lack of parameterization, and at the time of the community effort, we didn't really have a lot of experimentally validated parameters. So at this point, we are thinking how we could go forward in terms of developing small kinetic toy models to showcase interoperability and encourage people to join us in this effort. So at this point, we are, on Monday, we are submitting to peer review the first long paper that is in the bioRxiv server. And we have started drafting the second one that is mostly focused on this analysis and modeling ecosystem. I put the focus mostly on the modeling aspects, but there is also in parallel work that has been done in omics data and patients data analysis. So to wrap up, the focus of the community is on the mechanistic details where possible. So we like process description and we also like SBGN standards, but we are also open to collaborating with people who are not using this type of notations, for example, wiki pathways and activity flow, and we are trying to put effort in order to harmonize content and create crosstalks between different platforms. We also focus on transparency, interoperability. We are successful in translating process description to activity like Boolean models. So this is done and it scales up nicely. Of course, these models need training and tuning. The rules are preliminary. And this pipeline is also compatible with reproducible analysis of Jupyter notebooks of other communities. The C files obtained help other people who are interested in more mechanistic aspects, pathway analysis, and less dynamical ones. And at the end, we also foster exchanges and communication in order to develop multiscale modeling with PhysiCell and PhysiBots. So at this point, we curate new mechanisms. We try to harmonize efforts, improve technical aspects, especially for format conversions that are not ideal for some of the formats. See if we can automatize some parts of the pipeline that are not fully automated yet. And we are at this point analyzing drug datasets and patients' data. And of course, we are open to collaborations and exchanges with other communities that have similar interests. So that was it. I would like to acknowledge the whole disease map community for the vibrant discussions, exchanges, and for all this effort. And thank you for attending, and I would be happy to receive any questions. Thank you.
Thank you. That was lovely. We certainly have time for some questions. I think there are probably many questions. We have a limited amount of time, but definitely time for some questions. Do I see a hand up, Jacob?
So you mentioned that you use standards. One of them is SBGN, I noticed. Can you tell a bit more about the standard? Is it actually a standard? Is it like a standardization effort? There's a difference?
Well, it depends. I mean, we encourage people to use the standard, but sometimes people, either because it's not always easy to use the standard, or because the tools, for example, Cell Designer is not fully compatible with SBGN, but now they're making efforts to improve things there. So sometimes you don't have a way of validating how precise SBGN your diagram is. And this is why after the groups are constructing their diagrams, we pass a technical control, and we correct along with the SBGN community things that do not seem coherent or that they do not adhere to the standard. Of course, for some things, even the standards have not, let's say, good answers to give. So it is really an enriching experience, especially when some of the mechanisms are not known, and discussions such as how do we represent things that are not straightforward, or things where there is a certain kind of uncertainty. How do we quantify uncertainty in the interactions? So we use the standard, we try to use the standard, but sometimes it's not possible to say that all graphs are 100% SBGN compatible.
Allow me to rephrase. I'm not concerned about deviation from some sort of specification.
Okay.
I'm more curious about like the definition. What's the standard development organization behind SBGN?
So I think there is a page and tutorials where there is a certain way of representing biological mechanisms. So there are, let's say, suggestions of how to represent a complex formation, a complex dissociation, how to represent catalysis, translation, state transition. There are so many different biological scenarios that you can encounter when you are trying to translate knowledge into a mechanistic diagram. And for all of these scenarios, or most of them, there are certain rules that are proposed as the most appropriate for representing this type of
and who's organizing this? This is my question is like, what's the process? A standard usually has a standard development organization, a proper standard like ISO or something, or IEEE or something like this. Who's behind that standard? And who are the contributing people?
Sorry, Jacob.
Yeah.
Okay, we should also let other people ask questions.
Okay.
Yes, this is the SBGN community. I can actually send you the link afterwards.
Okay.
So there is a community that publishes regularly, and I think it was firstly initiated by Nicolas Lenover, but now there are other people like Falk Schreiber, like Alexander Mazain, like Vasuntra Touré, Adrian Rouni, that they are now the coordinators. So it's a community like the combined community or other communities in the systems biology.
You answered
field.
the question. It's not a proper standard. You should use the term standardization rather than standard.
Okay.
So there was a question. There was a question in the chat, which is, have you been able to use this to identify missing information? Has your standardization and harmonization approach identified critical missing information?
There are a lot of missing information. At least at first, there were a lot of missing information because we had limited access to, there was no prior knowledge for SARS-CoV-2. So when you are starting building mechanisms, but for most of these things, you actually rely on SARS-CoV-1 or on different coronaviruses. It's not that the information is not there, but the information probably is not 100% pertinent or relevant to this type of virus. So we build the mechanisms based on prior knowledge and incorporating sometimes things that are, I don't know how to say, a bit more generic, and then try to see if in the course of time, literature was on in order to justify all these interactions. So most of the diagrams were COVID-19, were SARS-CoV-1, let's say, specific in the beginning. And for example, the Reactome platform released, the first diagram that they released was the SARS-CoV-1 replication cycle. And after 6 months, 8 months, they just released the SARS-CoV-2. So it is a process. It's not something that is done automatically or, let's say, rapidly. But missing information, yes, it's all over, I could say.
I certainly would have lots of questions I'd love to ask, but I don't want to monopolize the conversation. Other people, questions? There are many people on the call who are involved in these efforts, so I'd like to other requests.
So I would like to say that the theses that we hold every Friday are also open to public. So if people would like to discuss more or exchange more, there is a Slack channel and weekly theses that they are highly welcome to participate.
So we should put that information up on our wiki so that that's available to the community on our end.
Yes, and also people are also encouraged to communicate via email if they would like something. I'm available for exchanges.
Other comments or questions? I thought I saw some. I guess I was going to come back then. Just one quick question is, well, not quick, but to this issue of kinetic versus Boolean modeling. Mm-hmm. Because when you're talking about gene regulatory networks, and Boolean models can be quite productive. And if you're going to talk about viral production replication, the rate at which that occurs is important. Or if you're talking about interferon pathways. Mm-hmm. And so I wondered if you could tell us a little bit more about this balance between kinetic modeling and which is difficult because of identification of kinetic constants, and Boolean modeling, where you can do these very large networks. What's your experience so far with that?
So I will give a very honest question. So my expertise comes from the Boolean and the logical network. I'm part of Colomoto. I was in the lab of Dignity3. So this is where my expertise lies. And last years with the INRIA people, we have been trying to create tools for large scale push to go a little bit higher than 20 or 40 nodes, but to the scale of 100, 200, 300 nodes and a proper analysis. Regarding the kinetic part, we have some people in the community that they are experts, but I think that they needed some time, so the context was ready and the interoperability was there. So I think that what we did was to facilitate the way for people to use this context in kinetic modeling. So there are tools like SBML Squeezer. They are SBML graphs that are very detailed in process description, that are compatible with COPASI or other modeling simulators. Even Cell Designer could be used for kinetic modeling, even if it's not so popular. So the material is there. People who are eager to try are really encouraged and welcome. So for me, it would be a lack of engaged people in terms of pushing this forward. So we have been trying to make things interoperable and easier for kinetic modeling. So if you would like to try, please feel free to pick a mechanism and start working on it. But it's not my expertise. So as I have no expertise in kinetic modeling, I cannot develop a kinetic model from scratch. Yes. For me, it's more interesting in the regulatory part, and this is what I'm doing.
And I see, for example, we have Robert Stratford on the call, who's a specialist in pharmacokinetic and PBPK modeling. So that would give you additional, I don't know if you have a whole body PBPK model included yet, but that would be a whole other scale that could potentially be very important. It's easy to translate those kinds of models, even if they're embodied at the moment in another format. It's very easy to put them into an SBML format and then pull that into the work you're doing. So Robert, I see, I'm sure there are many other people who could be the same, but I see Robert at the corner, so I would suggest possible connection.
Thanks, James. Be happy to help with that.
We would be happy to to facilitate exchanges. The context that the content that we create is accessible through the GitLab, and people are encouraged to use it. We are not keeping anything behind closed walls or just for the members of the community. You just need to contact us. If you don't find what you would like, we will facilitate your access, and well, that's it.
Okay. So if I don't see any hands raised, I had one bit of business to attend to. Anna, if you could put the main links into the chat during the last few minutes of our meeting. There were a set of
I will stop my
thank you.
My sharing.
If there are other urgent questions, by all means, but there just was one item of business that we had that was deferred from last week. Thank you so much for that wonderful talk, and I'm sure you'll be hearing. You said you were already of course, very busy. You'll be even busier now with requests for conversations from the people on this call. So thank you so much.
Thank you very much. I think I will be sending you the links because I'm having some problems with Zoom. It's not very responsive. So if it's okay with you, I will be sending the links to James, and he can diffuse them to your mailing list.
Thank you so much. It was very, very important talk. The one item of business that I wanted to address today was a discussion that was brought up in the steering committee, which was the idea about about how to do crowdsourcing. Now, we've just heard a wonderful talk about how to do crowdsourcing in the world of networks and SPML. And we've heard some very interesting things that they're doing in terms of connecting that with multiscale modeling with Physicell. Of course, we developed a software package, Computational, that supports SPML execution. And there are other packages that are attempting to do that as well. But one of the general questions about crowdsourcing, and Anna talked about this being a volunteer effort, is how do you encourage volunteer efforts? And one of the suggestions that had been brought up, although it's not entirely costless, was that it might be worthwhile to think about some kind of contest or prize mechanism related to, again, this working group is not a COVID working group, although we talk a lot about COVID. It's a viral infection and immune response working group, and on other aspects of viral infection. And so there was a suggestion that we should think collectively about whether challenges and contests would be useful in this community, whether that's a good motivational effort. And so I wanted to, we won't have much time to talk about it, but I wanted to raise that in the public forum for people's thinking and suggestions. Anna, I don't know if you've had any consideration in that direction, and whether, if so, whether you've had experience that way.
So we encourage people to apply for local funds. For example, we applied for French national funds, and people from the Wiki Pathways, they did the same for the Netherlands. Some people did the same for the US. Some of these efforts were successful. So we have now 2 engineers that work full time, and some people that their work is dedicated to it. But if you see the amount of progress and work is really nothing compared to how much this community has achieved. We would like to go full scale and apply for a European call, as most of the institutions come from Europe. But during the COVID, a lot of things changed. For example, we had a very big ITN network call that would facilitate PhD students and also doing some winter schools or summer schools. But these calls were canceled. And the problem that we have mostly in this type of, let's say, endeavors on finding money is that until now, we didn't have a success case. And what people ask for bioinformatics or for computational, let's say, aspects is, well, what is your success case? Do you have a drug that you propose? Do you have a hypothesis that you can validate? They need some kind of proof that what you are developing is really worth the effort and the money. So I think that now, after almost 1 year, 10 months of work, we have a body of work that can justify the demand of funds, and also collaboration with other communities would strengthen this demand.
Okay, I'm afraid we are running up against our time limit. We try to stick very closely to our conclusion, and it's very late in Europe, so we've kept Anna up very late. Thank you so much for everyone's participation. I hope that people will be contacting this group individually, and I'm looking forward to discussing it also in the steering committee.
James, I will be sending you the links, so it will be easier for me, and please diffuse them to your mailing list. And thanks everyone for your questions and for your comments. It was really nice being with you. Thank you very much for the invitation. I enjoyed it.
Reinhard, do you have any closing words for today?
No, not really. I just wanted to make everybody aware that Jeff Morgan in the chat indicated that he would be interested in the crowdsourcing contest. So if somebody's is also interested in that, please get in touch with Jeff.
Okay, thank you, everyone. We look forward to seeing you next week. We look forward to hearing reports on the subgroup organization. And thank you so much again, Anna, for telling us about this amazing project.
Thank you very much.
See everyone next.
Have a nice day.
Thank you.
Bye.
Okay, goodbye.
Cheers.